Welcome to this explainer. Okay, we're diving straight in today. We are exploring an absolute microscopic battlefield right inside the human body. We're going to unpack the anatomy of a cellular hijacking, a process known clinically as neoplasia. Throughout this explainer, we're going to treat the rogue, transformed cell as our main antagonist. We'll uncover exactly how it operates, how it breaks the rules, and ultimately, how it survives. So this brings us to the core mystery we're unraveling today. Why on earth would a perfectly healthy normal cell just go rogue? I mean, what drives a cell, one literally designed to work in total harmony with your body, to completely turn against it and start operating with this entirely selfish, destructive agenda? Section 1. The anatomy of a tumor. You know what? When we throw around the word tumor in a medical context, we're usually talking about a neoplasm. Clinically speaking, a neoplasm is an abnormal mass of tissue where the growth vastly outpaces your normal tissues. It's totally uncoordinated. But honestly, the most terrifying part of this definition is the end. It persists. Even after the initial trigger that caused it is gone, it just keeps growing. It's completely unrelenting. And these masses are basically made up of two parts. The parenchyma, those are the actual rogue neoplastic cells, and the stroma, which is the supportive tissue around them. Now, to ground all this microscopic science in reality, let's look at the actual numbers. Because this isn't just some abstract biological concept, right? This is a reality that affects millions. Looking at the global data, the top offenders vary a bit by gender. For males, prostate cancer makes up about 29.3% of cases, followed by lung and colorectal cancers. And for females, breast cancer leads the pack at 29.7%, also followed by lung and colorectal. These numbers, they really highlight exactly why understanding this cellular enemy is absolutely crucial. Section 2, benign versus malignant. So how do we actually categorize these rogue cells? Well, it usually comes down to how aggressive they are. Benign tumors. These usually end with the suffix -oma, like a fibroma or an osteoma. They typically stick to one cell type, they stay neatly encapsulated in their own lane, and crucially, they do not invade other tissues. Malignant tumors, on the other hand, they are the real threat. We call these sarcomas if they start in connective tissue, or carcinomas if they come from epithelial tissue. They're highly invasive, they completely lack cellular differentiation, and they have that deadly ability to metastasize, meaning they spread. Just look at the stark physical contrast here. On the left, we've got a lipoma. That's a benign tumor of fat tissue. Notice how the cells look pretty normal, right? They're organized, they're uniform. It's basically a localized nuisance. But check out the right side. That's a liposarcoma, the malignant counterpart. It is chaotic, it's disorganized, it's aggressive. The visual contrast in cellular organization between a benign and a malignant tumor is just night and day. If you're a pathologist looking under a microscope, you're hunting for very specific clues of this villainy. You're looking for pleomorphism. That's where cells just wildly warp in size and shape. You look for hyperchromasia, which means the nuclei stain incredibly dark because they're absolutely bursting with mutated genetic material. There's also the 1-to-1 nucleocytoplasmic ratio. Normally, a nucleus takes up maybe a quarter of the cell, but here, it balloons up to take over almost the whole thing. You'll also see a complete loss of cellular polarity and abnormal, totally chaotic mitosis. They're dividing in a complete frenzy. Section 3. How cells go rogue. Let's contrast a normal, obedient cell with the rule-breaking superpowers of a cancer cell. Normal cells are, they're good citizens. They wait for growth factors to divide, they respect their neighbors through contact inhibition, and if they get damaged, they undergo apoptosis, which is basically programmed cell suicide for the good of the body. Neoplastic cells, they do not care. They are completely insensitive to growth inhibitors, they magically evade apoptosis, and they have this limitless, unending ability to replicate. They literally just don't know when to quit. So how exactly do they get these superpowers? It all comes down to non-lethal genetic damage in two critical types of genes. Think of a cell like a car. Proto-oncogenes are your gas pedal. They promote normal, healthy cell growth. Tumor suppressor genes, those are your brakes. They stop unneeded cell division. But when a proto-oncogene mutates into an oncogene, the gas pedal gets jammed all the way down. And when a tumor suppressor gene is inactivated, it's like the brakes have been totally cut. Think about the security checkpoints built into a normal cell cycle. Usually, if a cell's DNA is damaged, tumor suppressor genes, like P53 or RB, step in at these checkpoints to halt everything and trigger repairs or apoptosis. But in a neoplastic cell, mutations have basically taken out these security guards. The mutated cell completely bypasses the standard protocols, divides anyway, and passes its broken DNA straight to the next generation. Now here's an absolutely crucial point. A single mutation doesn't just instantly give you cancer. It requires a sequence of catastrophic failures. We call this the multi-step carcinogenesis theory. Step 1, you get DNA damage from environmental or genetic factors. Step 2, the cell's internal repair mechanisms completely fail to fix it. Step 3, this failure leads to the activation of those oncogenes and the inactivation of the tumor suppressor genes. And finally, step 4, the cell starts this unregulated clone expansion, multiplying out of control. And the pathways mapping out this disastrous process are fascinating. Whether the initial DNA damage comes from outside agents, chemicals, radiation, or viruses, or from inherited genetic mutations, the pathway ultimately converges. The failure of DNA repair leads to an altered genome, growth gets promoted, cell death is avoided, and boom, you get clone expansion. And as that clone expands, it picks up even more mutations, driving full tumor progression, and, eventually, a malignant neoplasm ready to invade. Section 4. Building new supply lines. Okay, so once a rogue cell multiplies into a small mass, it hits a major roadblock. It needs oxygen and nutrients if it wants to grow larger than a couple of millimeters. To survive, the tumor kicks off a process called angiogenesis, the creation of brand new blood vessels. The tumor literally hijacks the host body, tricking it into building dedicated supply lines just to feed the cancer. Normally, your tissues keep a really delicate balance between signals that stop blood vessel growth and signals that promote it. But the tumor completely tips the scales. It starts pumping out pro-angiogenic signaling molecules. The most famous one is called VEGF. This floods the area, causing nearby blood vessels to actually sprout and vascularize the tumor mass. Suddenly, the tumor has its own private nutrient pipeline, and unfortunately, a highway to spread. Section 5. Invasion and Metastasis. Once that tumor is vascularized and breaks its local boundaries, it uses the body's own biological highways to travel. Carcinomas usually favor the lymphatic system, hijacking your lymph vessels to colonize nearby lymph nodes. Sarcomas, though, tend to favor the blood, entering veins directly to be carried away. There's also transperitoneal spread, which is where the cancer just seeds itself across open body cavities. And when they travel, these rogue cells frequently target organs like the lungs, the liver, and the brain. But the actual journey of a spreading cancer cell is incredibly treacherous. It's a multi-step process. First, they detach by dropping a sort of cellular glue called E-cadarin. Second, they literally chew their way through the surrounding tissue using secreted enzymes to enter a vessel. That's called intravasation. Third, they travel through the bloodstream as a little clump, or embolus. And finally, they have to stick to a new vessel wall somewhere else, squeeze out into the new tissue, which is extravasation, and try to start a brand new colony. Now here's a pretty wild fact. This whole terrifying cascade, it's actually highly lethal for the cancer cell itself. The human bloodstream is a very hostile environment. Only about 1 in 10,000 of these rogue cells actually survives the trip to establish a distant colony. But here's the catch. Because tumors shed millions of cells, even that tiny survival rate is more than enough to be completely devastating. And that brings us to a really grave clinical reality. Roughly 30% of newly diagnosed patients with solid malignant tumors already have metastasis. And once a tumor has spread to distant sites, the chances of a total cure drop severely. This is exactly why understanding the cellular villain and catching it early before those supply lines and biological highways are fully hijacked is absolutely paramount to saving lives. We've seen how chemical damage and genetic flaws can kickstart this whole terrifying process. But I want to leave you with one final provocative thought. What if the initial trigger, the thing that cuts the brakes and jams the gas pedal, wasn't just a random genetic mutation or some environmental radiation? What if the architect of the hijacking was actually an invisible virus? Biological agents, including viruses like HPV and EBV, are actually responsible for at least 15% of all human tumors globally. It's a chilling reality of microbial carcinogenesis that just proves how unbelievably complex this microscopic battlefield truly is. Thanks so much for joining me on this explainer, and keep that curiosity focused.